So today we will be discussing about plant growth and development. Growth, what it means is generally increase in the size of the plant. So the achievement in the increase in the size of the plant is first achieved due to cell division. And once the cell divides, you get from one cell, two cells, 4 cells, 8 cells, 16 cells, and so on. As they increase in number, their size also increases. So any quantitative increase in the size of the plant or its organ is known as growth. Whereas when we talk about development, it means the entire change that a plant it undergoes life cycle. So basically, if you were to measure growth, there's a simple instrument called the arc oxynometer. The arc contains many scales, and there's a pointer at which one end there's a pulley. One end of the pulley has a small weight, and the end of the pulley is tied to the tip of the plant. So due to the movement in the pointer, we can measure growth. This type of measurement by growth is done using an instrument called arc oxynometer, or you can simply measure them by measuring the heights at regular intervals or even measuring the diameters. But this is an instrument which has been used to measure the growth in plants. So generally when we talk about growth in plants, it is affected by various factors, external as well as internal. Among the most important external factor is light, because without light, plant cannot survive. Because of light, the plant is able to perform photosynthesis, and due to photosynthesis, it produces the reserve food material, which is used in producing energy for its various metabolic activities. So basically, when we talk about light, it depends on how much a plant gets a light during its day. So depending on it, there's a term called photoperiodism. The photoperiodism, what it means is the amount of light that a plant gets during a day. So depending on this, growth is affected. The other factor that affects growth is temperature. So generally when we talk about temperature, you need an optimum range to have maximum growth in plants. But by this, you should also be very clear, because there are plants which are suited to survive at cold temperatures, or even plants which are suited to survive at warm temperatures. But if we generally talk about plants which are generally growing in optimum range of temperatures, and if you take these plants to colder climates or hotter climates, then their growth will decrease. So generally, if a temperature increases, growth will also obviously decrease. So what you need is optimum range of temperature, but it depends on the habitats to which plants are adapted. The other external factor is obviously water. Water is essential for the survival of plant, because water helps in performing various metabolic functions. Also, water is involved in maintaining turgidity of the cells. By turgidity, what it means is when a cell is in fully swollen condition, then it is able to perform its metabolic functions at maximum ability. So the turgidity of a cell is very important, and this turgidity is due to water. The last external factor is pollutants. Pollutants, generally what they do is decrease the growth of plants. So plants obviously grow better in cleaner environments. Now when we talk about internal factors, the first thing that comes to mind is hormones. As you all know, hormones are substances which help in controlling certain specific functions. And generally hormones are produced at one organ and they act on another organ. So when we talk about hormones in plants, there are 5 such categories. Auxins, gibberellins, cytokinins, gibberellins, cytokinins and abscisic acid. So the first hormones that we will be discussing today is auxins. It was made famous by a scientist called Wendt, who performed a simple experiment. Coleoptile is a tip of a plant. What he did was, the tips of the pot coleoptile, he cut them. These tips were transferred to an agar block. Then this agar block were then placed in the cut tips of the coleoptile. So generally what happens is, when the tip is intact, the plant or the coleoptile increases. But the moment this tip is cut, there is no increase in the length of the plant. But when we place the agar block on the cut end of the tip, this portion of the plant is started to grow. So from this experiment he concluded that there must be some substance in the tip which is responsible for the growth of the plant. He proved this by, when the tip was cut, the plant did not increase in size. But when the same tip was placed in an agar block, some substance from the tip must have passed on to the agar block. And when this agar block was placed on the plant tip, this substance is again passed to the cut tip and it increased in size. So later, the substance which was coming out of the tip was discovered as auxins. So auxins are generally hormones which help in increasing the length of the plant organs, especially the roots and the stems. Gibberellins are basically hormones produced by fungus. It is also obtained in higher plants. So gibberellins was first discovered by Sabuta and Sunuki. These Japanese scientists, what they found was that a fungus called Gibberella fujikori in the rice plants produced plants which were very tall in length. But these plants, they did not have any seeds. So this disease due to the fungus is also known as foolish seedling disease in plants. So gibberellins, what they do is they increase the length of the internodes. Generally, gibberellins are produced in the embryos of the plant stage, but most of the gibberellins are basically produced by the fungus Gibberella fujikori. So one of the most principal auxins is indole acetic acid. In gibberellins, there are about 100, so depending on their chemical configuration, it is classified as G1, G2, G3 and so on till G100. Cytokinins are the next group of plant hormones. It was discovered by Weber Land. Cytokinins are basically hormones which help in increasing cell division. So when a cell increases in size or the cell divides continuously, it's due to cytokinins. So obviously growth of the plant takes place. The next one is ethyl. This hormone, its activity in plants was discovered by Neljubov. So basically ethylene is the only plant hormone which is in the gaseous state, and ethylene is basically used in ripening of plants. Ripening of fruits. And ethylene is produced by the leaves and flowers. Abscisic acid was discovered by Waring. This is the only hormone which does not promote growth, whereas all the other four hormones will promote growth. Abscisic acid generally promotes seed dormancy. So these are the 5 hormones whose roles are important in the growth of the plants. So in the next class I will be discussing about photoperiodism and phytochrome.